Okay, at the beginning of Chapter 4, Part 2, we are looking at the different layers of membrane. We have the nucleus first, and then the endoplasmic reticulum, the rough and the smooth. So the rough has ribosomes. We looked before, we saw ribosomes are where proteins are made. They're in the structure where we read the nucleic acid order, we put on amino acids, we build up proteins. So the rough endoplasmic reticulum is where proteins are made. And these proteins are going to get shipped through this membrane system to the plasma membrane or out of the cell. Otherwise, you have free-floating ribosomes that are making all the proteins we need inside the cell. So there, we're making proteins, and those are going to go onto the membrane. Okay, and then after the rough endoplasmic reticulum synthesizes or makes a molecule, it sends it on through what are called vesicles. These little membrane-bound sacs with the products inside. So you've got rough endoplasmic reticulum and smooth. So smooth is more about producing lipids, including steroids like testosterone and estrogen. You'll see lots of smooth endoplasmic reticulum in cells that are important for also detoxifying. So like liver cells, your hepatocytes, they've got lots of smooth endoplasmic reticulum so they can detoxify drugs. The products of smooth endoplasmic reticulum are also sent off in a vesicle. And where those go is the Golgi. So endoplasmic reticulum, Golgi. That's kind of like the post office. It takes the packages in that have been made at the endoplasmic reticulum, and then it can refine them, it can put a sugar on it, it can package it, and ship it out. So then you've got vesicles coming in and leaving at the other side, going out. And those vesicles, they can go to the plasma membrane and send products out, for example, or they can go to other organelles. The next organelle we're going to look at are mitochondria. They are really interesting organelles. They make the cell's energy. So they make energy in the form of what's called ATP, adenosine triphosphate. And it's got this 3-phosphate bonded portion, and those bonds are very high energy. And when you break one of those bonds, it releases energy that the cell can use to do work. So that needs oxygen. That's why we breathe oxygen in. We'll look at that more next chapter. And they are very similar to bacteria. They have their own DNA, their own RNA, their own ribosomes. And they have 2 membranes. They have one like a bacteria, and on the outside of that, they have one that is like ours. So that's why we have the endosymbiote theory, where mitochondria and chloroplasts, they have bacterial structure. So they believe scientists, they found evidence that a larger eukaryotic cell took in these bacteria and used them to make energy. So far back in the ancestry, this occurred. Mitochondrial DNA is also really cool in that you get your mitochondria from your mom. So if you want to trace back your mitochondrial DNA, you can go back a lot further. So if they want to see where ancestors were from, like different continents, they can use mitochondrial DNA. So this shows the mitochondria with the outer and inner membranes, their own ribosomes, their own bacteria-like type plasmid DNA, and they do the processes of cellular respiration, making ATP. Okay, and ribosomes, we discussed these when we looked at the endoplasmic reticulum. Those make proteins from RNA. So you've got some that are free, just floating in the cytoplasm, and some that are bound to the endoplasmic reticulum. So those free ones, they make proteins for the cell, for the other organelles. Membrane bound, generally are going to go to the plasma membrane or exported. Peroxisomes. You've got membrane bound sacs with very strong enzymes inside. They detoxify harmful substances. So what they do is they neutralize free radicals, these like highly reactive chemicals. Their electrons are unpaired, so they're looking to make changes, make reactions. And what they do is first, they have these enzymes that convert the free radicals into H2O2, hydrogen peroxide, which is also toxic. So then they have enzymes that convert that into water and oxygen. So when you put hydrogen peroxide on a wound, that's what's happening when it bubbles. You're making oxygen. Lysosomes, these are like the stomachs of our cells. They have enzymes in there that break things down. So like anytime you've got old organelles, your white blood cells take in bacteria, they bring those in, they merge them with the lysosomes, and everything gets broken down. If you have cells that are overcrowding in an area, they'll release their digestive enzymes, the lysosomes will break open, and you'll have this autolysis, this self-breakdown. Auto is self, lysis is breakdown. So here you can see the green areas are where lysosomes are active. And then cytoskeleton. We discussed when looking at the types of proteins that there are different filaments, these protein structures inside that give the cell its shape. That's the cytoskeleton. Centrosomes and centrioles. These are going to be important for cell division. They're going to organize what's called the mitotic spindle fiber. We'll look at that next week. They go to each side of the cell and they help us get exact copies of DNA into the two new cells. So here you can see them. And this is a nice microscopic view. They've got this lovely 9x3 structure. So everything we just described is part of the cytoplasm. That's the inside of the cell with the fluid, the cytocell, the solution, the water, the organelles, and then inclusions. These are going to be used for storage generally. Now that we've seen what's inside of the cell, let's look at what's on the outside of some. Some specialized cells will have cellular extensions, where their plasma membrane, because of the cytoskeleton, gives it a different shape for different purposes. So you've got cilia and flagella. So cilia are like these hair-like substances that they move in a wave-like motion, and they push things across the surface of the cell membrane. Whereas the flagella, you'll just have one long extension that propels the cell itself. So here's cilia, so like lining your respiratory tract, the uterine ducts, you've got cilia that move substances across the surface, whether it be mucus, so we can swallow it, or it can move the egg down the fallopian tube. See the mucus moving. When it's really cold outside, your cilia and your nose get inhibited. They're not able to work as properly. So that's why the mucus drips out of your nose. The cilia is not pushing it back. And then microvilli. So in some areas, we want to have lots of surface area, more space for plasma membrane, for the proteins on the outside, so that we can absorb more of our nutrients, like in our digestive tract. And next, vacuoles. A vacuole is a storage unit. So in different cells, you can see different types of storage units, different vacuoles. So adipocytes, these are our fat cells in our body. Basically, the whole cell is a fat storage vacuole. The nucleus gets pushed to the side because the fat takes up all of the cell. In plants, you have a central vacuole with lots of water in it. That helps give the plants their shape. Generally, in animal cells, you'll actually see smaller vacuoles. Plants have larger but fewer per cell, but they both have them. So everything that we've looked at so far, both plant and animal cells share as organelles. They're both eukaryotes. They are quite similar. However, there are a couple organelles that are specific to plants, and we'll look at those next. So first is the cell wall. Outside the plasma membrane, cell walls exist, and they're very structured. They're very rigid. They give plants their shape. So if you looked at a microscopic view of a plant, you'd see these rectangle square cells right next to each other. And as long as the vacuole has water in there, the plasma membrane goes right to the edge of that cell wall. And the other portion is chloroplasts. So you've got all these green circles here. Those are chloroplasts, and inside there you've got the reactions that are occurring to make energy with photosynthesis. We'll look at that more next time. So something to remember, we are not only our own cells, we've got lots of prokaryotic cells inside us too. They can influence what we do, and it's important for us to understand them as well.